honor for me to be inducted as a fellow of this highly esteemed academy, and I would like to thank the academy and the organizers for giving me an opportunity to present our research work. Today I would like to discuss a part of our research, that how nucleic acids can synthesize their own ligands and that show promising biological applications. So, to begin with, I would like to give a brief outline of our group research activities. One of the major areas of research in our group is targeting nucleic acids by small molecules, and we also use nucleic acids, particularly DNA, to devise biomolecular devices. For instance, very recently we have used a DNA to construct an artificial ion channel that can transport potassium ion, that can transport selectively a particular ion, that is potassium ion, across the biological membranes. And in addition, we use DNA and RNA bases as the molecular building blocks to construct biomimetic functional systems. In addition, as a synthetic organic chemist, we also synthesize new molecules, and for that we develop new synthetic methods and new catalysts, and those compounds we use for targeting nucleic acid secondary structures. So, today I would like to talk about a part of our research, that is targeting non-canonical nucleic acid DNA secondary structures. We all know DNA is mostly double helical, but it can also adopt other higher order secondary structures. For instance, a G-rich DNA sequence can adopt a G quadruplex structure, and its complementary series DNA sequences can also adopt 4-stranded secondary structures called I motifs under acidic conditions, and these structures are called I motifs as they contain intercalated cytosine-cytosine base pairing due to hemiprotonation of one of the cytosines under acidic conditions, and these G quadruplex structures contain this G quartet as a basic feature, which contains the square planar arrangement of 4 guanines that are linked together by non-canonical, that is Hookstein type of hydrogen bonding. And these G and C rich DNA sequences are present in the promoter regions of several oncogenes, and it has been shown that small molecules can bind and stabilize these structures and modulate transcription in cancer cells. Therefore, small molecules that can bind and stabilize these DNA quadruplexes are considered as potential anti-cancer targets. And in recent times, the I motifs have also emerged as potential anti-cancer targets. So, in our lab, we have synthesized and designed different classes of compounds like heteroaromatic compounds, molecules inspired from nucleosides and peptidomimetics for selectively targeting one of the DNA secondary structures. For instance, one of the peptidomimetics, this is a natural product like a distamycin analog. Distamycin is known to bind both double helical DNA and 4-stranded non-canonical structure, but this compound is highly selective for the non-canonical DNA secondary structure and shows promising anti-cancer activities. And very recently we have also synthesized a PNA mimic by replacing the natural nucleobases with a thiazole ring with an amino group. So, here the idea was to mimic the nucleic acid and protein interactions. So, this is like a short peptide like molecule that can selectively bind a DNA quadruplex structure in the cellular system. However, the rational design and synthesis of selective ligands is always difficult and time consuming, and most of the reported compounds are not suitable for advanced clinical trials because they are not soluble in water, they are poorly soluble in water, they lack membrane permeability, and most of the compounds are also synthesized using multi-steps. So, we thought of synthesizing molecules by using simple synthetic steps. Next, it is important to design ligands that show not only selectivity for a 4-stranded DNA quadruplex structure over the double helical DNA, which is highly abundant in the human genome, but it is also important to develop ligands that show selectivity between different higher order secondary structures. And next, the ligands should be designed in such a way that they could be easily detectable in the cellular system. And finally, the most important question is whether a ligand really targets the secondary structure and then alters the transcription. So, the insights into the mechanism of regulation of gene expression by a small molecule ligand needs to be developed. Considering all these aspects, we thought of using nucleic acids themselves in a chemical reaction and synthesize their own selective binders. So, this is a classical process of identifying drug-like candidates. So, the classical process to identify or discover a drug molecules begins with the synthesis of small molecule libraries and followed by their biophysical or biological screening to identify a few active compounds. Most of these compounds are synthesized, are individually synthesized in the lab using large quantities of organic solvents and reagents. So, thereby making the library synthesis costly and time consuming. So, we thought of combining the first two steps like synthesis and screening to one step by using the target directed synthesis. So, in this approach, small molecule fragments binds to DNA or RNA targets or any biological target and then come into close proximity. So, they bind to the adjacent sites of a target and then comes into proximity and then undergo an irreversible reaction to provide products that can strongly bind to the biological target. Since the biological target selects its own fragments, so the resulting products are more selective for that particular target. So, in this way we can synthesize and also screen the compounds in a single step. So, in our approach we have taken two different quadruplexes and their corresponding I motifs, and these DNA sequences are present in the promoter regions of c-MYC and BCL2 oncogenes that play key role in cancer progression. And we have also taken like a double helical DNA and a single stranded DNA as the control templates. So, in our example, we have taken thiol containing DNA sequences and immobilized on gold coated magnetic nanoparticles. So, our idea was these DNA nanotemplates would accelerate the rate of chemical reaction that just we have seen here, that it would, if we use a DNA nanotemplate, then it would accelerate the rate of reaction by providing a large surface area with more number of localized DNA templates, and moreover the DNA nanotemplates could be easily recovered and reused for subsequent reactions. So, in our approach we have synthesized a mixture of a pool of alkyne and azide fragments and then incubated with the quadruplex nano template. And then after giving sufficient time, it was almost 6 days, we have used an external magnet to isolate the DNA bound ligands, and that is we have isolated the newly formed products along with the DNA nanotemplates, and the unreacted starting materials remained in the supernatant. And then this ligand bound DNA was further heated to identify the products which are characterized by using HPLC and mass. And as I have mentioned that we could also recover and reuse the DNA nanotemplate. So, we have taken a mixture of like 4 compounds, 4 different alkynes and 17 different azides that could provide us 136 possible compounds including syn and anti-triazoles, and that are likely to interact with the DNA targets by using pipette stacking and electrostatic interactions. The same library was also used for the I motifs, and since I motifs are stable under acidic condition, so this reaction was carried out at low pH. So, by using HPLC and mass, we could identify 6 different compounds by which were formed by using the make I motif as the nano template, and then we also found out that only 5 compounds were obtained by the control duplex DNA. So, the product that was selectively obtained by the c-MYC I motif and not by the duplex DNA was considered as a selective lead for the c-MYC I motif, and similarly we could identify a selective lead for each of these 4 DNA secondary structures, that is c-MYC quadruplex and its corresponding I motif, and BCL2 quadruplex and I motifs. So, without synthesizing all these 136 compounds, we could identify 4 selective leads, and we have also did not do any biophysical screening to identify which is the high affinity binders. So, next we have taken all these 4 compounds to cellular system. I would like to give one example of a ligand that was obtained by the c-MYC quadruplex. Like fluorescence microscopy revealed that this ligand is cell membrane permeable and mostly localizes in the cellular nucleus, and then we have also carried out immunocytochemistry using BG4 antibody. BG4 binds to the G quadruplex structures and produces red dots, which indicates the presence of G quadruplex structures in the cells, and ligand treated cells produced more number of red dots, which indicates that the ligand co-localizes with BG4 and also produces also induces the G quadruplex structures in the cell. So, this study indicates that the ligand can bind to G quadruplex structure and also induce their formation in the cancer cells. So, next we have studied its effect in the regulation of c-MYC gene expression, that is this ligand could reduce the c-MYC expression in a dose dependent manner. We have carried out both qRT-PCR and Western blot, which shows that it reduces the mRNA and protein level in a dose dependent manner without reducing the GAPDS, the control gene expression, and it can also selectively kill the cancer cells and not the normal cells. So, from this study we know that this ligand can downregulate c-MYC transcription in cancer cells, but next, whether this ligand really reduces the c-MYC expression by directly targeting the c-MYC quadruplex structure. So, for that we have carried out an exon specific assay. In this assay we have taken a CA46 Burkitt's lymphoma cell line. There is a translocation between c-MYC gene of chromosome 8 and immunoglobulin gene of chromosome 14, and due to this translocation exon 2 is translocated to the immunoglobulin gene, and therefore the exon 2 is not present in the c-MYC gene. A G quadruplex ligand would only reduce the exon 1 transcription without affecting the exon 2 expression, and our results show that the c-MYC quadruplex lead could selectively reduce the exon 1 expression without altering the exon 2 expression, and this assay validates that the reduced expression of c-MYC expression is due to ligand induced stabilization of the 4-stranded secondary structure. So, next we thought of using a biomimetic template to identify G quadruplex ligand instead of using the biomolecule itself. So, in the beginning I have discussed that a G-rich sequence can adopt this G quadruplex structures in the presence of metal ions like potassium, and it contains this G quartet like assembly. So, being a synthetic chemist, we also synthesize nucleoside derivatives and study their self assembly, and we have shown that a simple guanosine derivative with boronic acids can form this G quartet like assembly, and that can mimic the topology of a G quadruplex structure. So, we thought to use this hydrogel assembly that contains G quartet like structures to promote this cycloaddition. So, our idea was the resulting products would strengthen the gel network, they can bind and stabilize the G quartet assembly and then drive the cycloaddition. And quite remarkably, like the semic quadruplex, this gel assembly could also synthesize this compound in a regioselective manner, and thus we could use a cost effective material like a gel system than the quadruplex itself, and this can be used for large scale synthesis of the ligands. And further, this study would also inspire other biomimetic synthons for other biomolecular targets. So, next we thought of using this strategy for RNA, which is more dynamic in structure. So, we have used TAR RNA, which is present in the HIV-1 genome, and this TAR RNA can adopt this type of secondary structure, a stable hairpin structure containing UCU bulge, a 3-base bulge, and this hexanucleotide loop region. So, it has been shown the binding of a TAT peptide with TAR RNA is promotes the viral replication. So, therefore, TAR RNA has been targeted by ligands to inhibit TAT-TAR interactions and thus viral replications. So, considering the dynamic folding topology of the RNA structures, we thought in situ cycloaddition could be a promising strategy to identify selective ligands for this RNA structures. So, we have used TAR RNA as the target and a TAR RNA without this bulge region as a control template, and we have also taken a corresponding TAR DNA. So, we have synthesized a mixture of compounds, and then in this case the reaction is essentially same fragments with the biomolecular target and a buffer that is suitable for the biomolecule to be stable. And in this case we have used biotin TAR RNA and used streptavidin based magnetic beads to isolate the RNA as well as the newly formed ligands. So, by using HPLC and mass we could identify 3 different lead compounds, and one of them is selective for the TAR RNA, and further this cycloaddition, this in situ reaction also provided insight into the binding mode of the ligand to the secondary structure. That is, this compound which was formed by TAR RNA was not formed by the TAR RNA without the bulge region. So, which suggests that this ligand possibly binds to the bulge region of the target, and the TDS, that is target directed synthetic results, are also consistent with the biophysical screening. That is, the compound which was selectively obtained, that is a thiazole based peptidomimetic containing this flexible CST units, was also found to be selective for the TAR RNA that binds to the TAR RNA with a nanomolar binding affinity and 1-to-1 stoichiometry. And then we have also carried out a Fred based assay to find out that this ligand binds to the TAR RNA and also displaces the TAT peptide, and further molecular modeling suggested that the ligand binds to the bulge region of the TAR RNA. So, thus this is a rapid method to develop a new class of peptidomimetics that can bind to the TAR RNA, and the molecules that bind to the TAR RNA and inhibit TAT-TAR interactions are considered as potential antiviral agents. Yeah, so thus I have shown you that in situ cycloaddition can be used to develop selective ligands for DNA secondary structures like chick quadruplex, I motifs, and for RNA secondary structure, and we have also used a biomimetic synthon to synthesize ligands in large scales. So, the advantage, the method reduces the time required for the conventional chemical synthesis and the biophysical screening, thus it bridges the gap between synthesis and screening, and the products can be isolated easily, and also it also enables easy identification and isolation of the compounds. It prevents the wastage of expensive commercial DNA targets, and moreover the ligands show high binding affinity and as well as biological activity. So, at present we are using this method for new and challenging targets like COVID-19 virus. There are different RNA structures which are present in the RNA that we are currently working on, and in addition we have been trying to study small molecule nucleic acid interactions for biomolecular devices that I have already shown. I do not have time that to devise artificial ion channels, and finally I would like to thank my research scholars for their hard work and cooperation, funding agencies for financial support, and I would like to thank you all for your time.
What's the Uh, I see that triazole is one of the group that is present in most of your most of your work.
Yes.
So, what is the special observations you made with respect to the presence of triazole in all of these chemistries?
Actually, as we are using the in situ cycloaddition between azides and alkynes, so it is expected that the products would be triazole. Yes, so it's a bioorthogonal reaction. We are using this because these starting materials are biocompatible, so that it won't react with the biomolecules. So that is the reason we are using that reaction. I have also mentioned at the end, which I did not discuss, that we are also trying to identify new bioorthogonal reactions, so that instead of only triazole, of course that is nice because it's a peptide mimic, amide bond mimic, but we can also use different reactions, so that there will be more diverse chemical structure. For instance, you can use like a one thiazolidine-dione aldol reaction, where you can get easily the compounds. You can make those handles easily, and the library synthesis should not be that difficult.
Hi, that was a very interesting talk. Jyotirmayee, I was wondering, you said in your talk that some of these ligands induce G quadruplexes in the cells?
Yes, 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 yes. Not only in the cells, we have also did biophysical studies where we have taken a G-rich sequence. And if we just take a G-rich sequence, it is in vitro, then it does not form a G quadruplex structure, it is a random coil. But only when we anneal with potassium, then only it forms a G quadruplex structure. Even if we add only potassium in the real time, also we do not see a structure. But
No,
some,
in vitro,
yes, that is what, but some of the ligands that can induce the formation of structures, particularly this ligand, if you do even in vitro, just G-rich DNA sequence and this, then you can see a nice CD, you can nice structure, you can see a Fret based assay where you can find that it indeed induces, and in the cells also it induces, because BG4 is an antibody, it's a G4 specific antibody, so it binds to G quadruplex structures. So when we add BG4 and if we see that red dot, so it shows the presence of G quadruplex structures in the cells. But if you treat ligands, not all ligands, if you treat other ligands that do not induce, then we see only those many red dots before and after, only a colocalization. But in this case, what we have seen is more number of G quadruplex structures, means more number of red dots, which indicating that it can induce the formation of structures in the cells.
All right.
For the fluorescence microscopy, which software you use for that?
It was just not so whatever is there in the inbuilt. We haven't used anything. You just take and you get the image.
I suggest we take any further questions during the lunch interval.